And the next speaker, Mihály Vöröslakos, Dr. Vöröslakos from NYU. Are you with us?
Yes, good morning. Thank you for organizing this webinar and inviting me. So today I'm going to talk about how we can achieve spatially and temporally targeted neuromodulation in rats. Next slide. So as I'm sure you're all familiar, but like just to recap a bit how physiological effects depend on the dose of stimulation. So transcranial direct current stimulation will induce a diffuse electric field inside the brain. So as you can see in this picture, the electric fields are highest underneath the stimulation electrodes, shown in yellow, and then it drops with the distance. And in the middle of the brain, there is barely any change induced by the stimulation. So, and why the electric field is so important? Because we know from many studies that the neurons are influenced by the electric field. So the higher the electric field, the more neurons will be affected, and the effect at an individual neuron level will be also higher. So we can, as Dr. Discussant mentioned, that we can have a higher change in membrane potential. Next one. And besides of the electric field, another important factor that many others mentioned is the orientation of neurons relative to the electric field. So different orientations can lead to soma depolarization that you can see on the left, or hyperpolarization that you can see on the right, or no change in the membrane potential. So next one. But unfortunately, the spatial focality of transcranial direct current stimulation is relatively poor. And focality refers to the ability to restrict the effects to a target region while minimizing the currents in non-target areas. And so, for example, you can see this black circle on the right side that would refer to a focal effect, only have an influence on neuronal activity at this particular region of the brain. So one solution to increase the focality of the stimulation is to use multi-electrode stimulation methods. And then the more stimulation electrodes we can use, the greater the potential to generate spatially restricted effects. And so new engineering solutions have been proposed. One of them is the high definition TDCS, or the other one is the temporal interference stimulation. So, and then during my PhD, we also developed a method called intersectional short pulse stimulation. And then you can see on the bottom right, the idea that we use 3 electrode pairs to distribute currents by temporal multiplexing. And then the electric field vectors add together, so ISP may allow higher overall stimulation intensities. So, and what is this ISP? We just turn on and off the stimulation electrode pairs, one after the other. And then in order to distribute the currents, we just change this configuration. So, and then to test whether ISP can modulate the ongoing brain activity, we apply the stimulation targeting the ipsilateral or contralateral hippocampus that you can see on the bottom left side of this slide. So next one. So, and then to measure the neuronal activity, we inserted a silicon probe into each hemisphere and recorded single unit activity in 8 anesthetized and 1 freely moving rats. And then on the right side, you can see our population results. So 55 neurons show the laterality effect. And then we measure the effect of stimulation as the false change in firing rate, where the baseline firing rate is 1, which is shown by the dashed line. And you can see that there were neurons that increased their firing rate. So it's above this dashed line, or some neurons decrease their firing rate, which is below this dashed line. So next one. So, and the reason for this is because ipsilateral ISP under the anode, which you can see now on the left side of this plot, will inhibit hippocampal neurons, and under the cathode, it will excite hippocampal neurons. And when we have this mirror configuration for stimulation and recording, what we can, I hope you can appreciate that we can record simultaneously soma D and hyperpolarizing effects. Next one. And so to further investigate this, whether single unit response reverse with polarity and scale with intensity, I implanted a freely moving rats with silicon probe in the hippocampus that you can see on the top. And then now, instead of using this mirror configuration, I apply different stimulation intensities and polarities. So 4 seconds stimulation was followed by 4 seconds no stimulation, as you can see on the bottom left side. And then the box plots shows the median firing rate of the significantly modulated neurons. And as you can see, the higher the stimulation intensity, the bigger the change in firing rate. And then this change is reversed by polarity. And thanks to these high density recordings, the single units, we were able to categorize them into putative pyramidal units or interneurons based on their waveform metrics. And pyramidal cells are shown here by the red triangles and interneurons by these circles. And as you can see, even at the lowest intensity, as Dr. Bigson emphasized, that this how low you can go, even at the lowest intensity, we can have a handful of neurons that is significantly modulated by stimulation. So, and then one of the big questions that I hope we can address today, does this translate into behavioral responses, and where to go next? Thank you.
Thank you very much.